now the floor is to Maria Chait from the Ear Institute at University College London, which deserves like maybe even more thanks than the other speakers, because she's heroically speaking from a COVID household. So thanks a lot for being here. And yeah, the floor is yours.
Oh, right. Oops, sorry, just a second. I'm just trying to optimize my screen. Okay. Can you hear me?
Yep. Very
awesome.
clear.
Okay. So yeah, thank you very much for organizing this. I think that, you know, for a long time, I've been talking to a lot of people about the fact that there's these two subfields that I consider myself being sort of part of, that I wish, you know, integrated more. And yeah, so this is a great initiative to start off this process. I'm also a little bit embarrassed because we were slowly climbing up the hierarchy of, you know, of complexity in terms of the stimulus that we're talking about. We started with sort of abstract sounds with Tristan, and we moved all the way up to language. And now I'm going to be plopping us all the way back down the same hierarchy, looking at abstract sounds. And in general, this topic that I'll talk about is part of a bigger research effort in my lab, which is looking at the auditory system as the brain's early warning system. So the idea is that the auditory sense, the fact that our ears are always open and receive information from all around us all the time, serves this purpose of monitoring the environment for potentially behaviorally important events and bringing these events to our attention. So the kinds of questions that we're interested in tackling in the lab relates to understanding what information is being tracked by the auditory system when we just, you know, exist in the world, and also what kind of events capture attention. So unlike the I guess the previous talks, I mean, here in almost all of the experiments that I'll describe, the sounds are irrelevant to the task. So participants are either listening passively or performing some sort of incidental task. And we're looking at how representation of this unfolding stimulus builds up in their brain. And the statement that, you know, like this all starts with, and one that I don't need to convince anybody here of, is that the auditory brain is exquisitely sensitive to patterns in sound. We know that intuitively from listening in the world. And there is a huge literature in the MMN field and others that has formally demonstrated this. And this is because, I mean, this sensitivity is hypothesized to arise from the fact that many behaviorally relevant sounds, so sounds that we're potentially evolved to be sensitive to, contain predictable patterns because of the sort of limits of the physical bodies that generate them. So for example, we can listen to this sort of clip of the soundscape of the African savanna here. So the idea is that these sort of predictable sounds are all around us, and we use our sensitivity to structure to identify what they are, but also this detection of regularity helps us listen to environments like this by making us more efficient at processing events that we expect, and also making us faster at detecting changes. And my, you know, out of the natural world and back into the lab, the example that I usually use to illustrate this is the example that is shown here. So these are just like, I'm going to be talking about abstract sound sequences. So the x-axis is time and the y-axis is frequency. And these little rectangles are 50 millisecond tone pips that are arranged in a particular way. They have no significance in the world. We can arrange them in a regularly repeating pattern like what is shown here. And we can also, and we can also arrange the same tones in some sort of random pattern, but otherwise being spectrally identical. And what you might notice also is if you look carefully is that there's a deviant in each sequence. So there's just a single tone pip that sticks out, that is away from the frequency range that it's occupied by the sequence. I mean, there it is. And I can have you listen to it. So these will be sounds that you've never heard before. And let's see if you hear the deviant tone in this sequence. So normally I actually stop and have people tell me if they had or not. So here I'll skip that part. And normally the very few people hear the deviant. However, if I embed the deviant in the same sequence, so these are the same sounds, but they're now arranged in a regular structure, it sounds like this, where the deviant is much more obvious to detect. And this is remarkable because the regular structure is one that you've never experienced before, yet the deviant immediately pops out, suggesting that your brain had within the span of a second or two, discovered the pattern, learned to predict what is going to come next sufficiently well to respond to this outlying event. And people in the lab indeed behave in the same way. So their sensitivity is much larger to these deviants in regular relative to random sequences. And the same is the case when we record their brain responses while they're listening to these sounds and watching movies, so when they're entirely distracted. So what is shown in light blue here is the response to the deviant in the regular sequences, and the orange trace is a response to the deviant in the random sequences. The difference between the two is much less dramatic than the behavioral difference, but it is present, and we can quantify the response in terms of sort of the main response to deviance, to frequency deviance, and the particular portion of the response that relates to detecting an unexpected tone within a highly predictable sequence. So this effect of regularity on the deviant response. They engage slightly different channels. And when we look at source localization, I mean, this is EEG, so it should be taken with a huge grain of salt. The standard sort of main effect of outlier engages the standard temporal or frontal network that Tristan has been talking about, that Lucia has been talking about, and I'll return to in a minute. And this effect of regularity, so this extra bit of the response that relates to detection of deviation from this established pattern, appears to engage more frontal sources that are distinct from this network, and which encompass the orbitofrontal gyrus and the temporal pole. But now the question is, and that has been sort of engaging work in my lab for quite a long time, is how do we detect the structure in these rapidly unfolding sequences? And for a while, so starting in the last 5 years or so, we've been using a paradigm that involves presenting people these specifically structured tone pip sequences that would either contain or not contain regularities, and measure their brain responses to these sequences or transition between different states of regularity. And the general finding in this work suggests that, so this shows you an MEG response in a distracted participant listening to this random tone pip sequence, so a bit like the ones that I've played you already. And the response when we introduce a regular structure, so when we introduce a transition from random to regular, which sounds like this. So these are rapid sound sequences. The regularity emerges quite quickly, but is very easy to detect. So this transition is ubiquitously across a lot of experiments with different types of stimuli, is associated with this pattern of response of divergence between the random and the regular sequence. It's associated with an increase in the sustained response. I'll devote some more time in a bit later talking about specifically what this type of divergence tells us about what the brain is doing. Oops, sorry. I've had the pleasure of listening to these things twice. And the idea what this sort of demonstrates is that the brain automatically searches for and detects regular structure and sequences even when you are not seeking them. And the point of detection, so the latency at which these things diverge, happens really quickly. So in fact, as quickly as is predicted by an ideal observer that can sort of track the statistics of these stimuli. Importantly, we see this type of divergence across, actually before I go to that, when we try to localize which brain areas underlie this process of discovering irregularity, the network that pops out is one that is very similar to the one that Tristan has been discussing in his talk, and actually that Luthia has been discussing in her talk as well. And that is this bilateral auditory cortex, frontal IFG, and hippocampus, which suggests that this ubiquitous network that has been implicated in detecting deviance, or in the mismatch negativity type response, is also the same network that gives rise to these processes that underlie the discovery of structure. And like I said, and there's no brain areas that prefer random over regular. So the contrast of random larger than regular yields nothing. This type of difference, by the way, of larger responses to regular versus random is also seen in the bold response in fMRI, which suggests that, there is something is responding more to these regular sequences. And like I said, we see this ubiquitously across different types of regularity. So we can see this difference between regular and random using really complex scenes of multiple streams that are either temporally regular or random. And we can also see it when we look at stochastic sequences. So these are sequences that don't have a precisely repeatable pattern, but where the transition is associated with a change in statistics that makes the sequence post-change more predictable. So in this example, we start off with a random sequence of tone pips, and then we transition to another random sequence of tone pips, but where the distribution is structured. So here we're sampling these tone pips from the high frequencies and low frequencies only. So these patterns sound like this. They're quite hard to hear out. I think I have another example, or maybe I don't. This is what the EEG response looks like to this no change random sequence. So you'll notice it's noisier than the previous data I've shown you. That's what happens when you go from the luxury of MEG to, the noisy EEG. But again, in distracted movie watching individuals, we again observe this response that's associated with the discovery of structure. And we hypothesize that what it reflects is some sort of form of model establishment, so an establishment of a predictive model. And so the next question would be, what information is the brain tracking? So what information does the emergence of this response reflect? And to do that, we used two types of sequences that are depicted here. So they're both starting off as a random sequence. So we're pulling from a pool of 20 frequencies randomly. And there's transitions to a structured sequence. So in the top one, so this R20 to R5, the transition is to 5. So we transition to a pattern where we randomly pull 5 frequencies from the pool. And the bottom one, and it sounds like this. In the bottom one, so this R20 to R10M, we transition from, again, this random pattern of 20 tones to a random pattern of 10 tones, but where we pull from the middle frequency range, and it sounds like that. So we chose them for several reasons. So first, they are statistically different from each other, and we can make different predictions about what statistics the brain might be tracking. They're both behaviorally yielding quite similar performance. So when we have people actively respond to these transitions, people are equally, well, they're a little bit slower in the top, like the R5 condition relative to the R10 condition. The D prime is about the same. So suggests that so behaviorally, they're more or less comparable. But importantly, statistically, we can make different predictions about what brain responses these two might yield. And for that purpose, we've been using a Bayesian sequential prediction model that has been kind of proposed and implemented by our collaborators in previous work. And what this model is doing is through exposure to this unfolding tone tip sequence, it sequentially generates a predictive distribution of the frequency of the upcoming tone. So we can feed it with some priors, so what it believes the initial distribution to be, and then it sort of refines this distribution over time as it encounters more and more information. And specifically, the model tracks. So it's limited to be more in the kind of ecologically plausible by implementing a memory constant and noise and so on. And what it tracks is it tracks two possible parameters. So it tracks change probability, which is the probability that there's been at least one change in the sequence. And it can also track precision. So precision would be a measure of how precise the predictive distribution is. So it's a measure of the width of the predictive distribution. And interestingly, I mean, this model has very different predictions for these two sound sequences that we're using, depending on. So if we believe that the model, I forgot to say that we are manipulating two things. We're manipulating the prior, so whether the model thinks that that the world is drawn from one distribution, so whether the world is initially a single channel, or whether the world is actually several concurrent channels, sort of a multiple distribution. And that is a model of how we think people are listening, whether they're listening holistically to these sequences, to these tone patterns, or whether they're listening to them in a channel by channel basis. And this model produces different predictions for what we might observe in the brain, depending on whether we're hypothesizing that the model has a single prior, so the single distribution, or a multi-prior, and whether we're hypothesizing that the model might be tracking change probability versus precision. So what is plotting here are these outputs. So black is the no change condition, blue is this condition, the R20M, and gray is this condition, so R20F. So you can see that there are different predictions that it makes. So what do the EEG data for these conditions look like? And this is what is plotted here. So again, these are naive people listening to these sequences, and it's clear that that a model that is outputting precision as its output, and one that has a multi-channel prior, suggesting that listeners are listening to these things via separate channels, is the one that provides the best account of what we see in the brain. The other ones are actually producing opposite patterns to what we are seeing here. And we're using these sorts of evidence to support our hypothesis that the EEG response tracks the precision of the predictive distribution. So basically it reflects the emergence of confidence in what we might expect will happen next. What is important to say about this, though, is that this effect is independent of attentional capture. There's been a lot of discussion in the literature about whether predictability attracts attention in an automatic manner. And there's a good argument to be made about the fact that predictable objects in the world are probably meaningful and hence should attract our attention in some way. And in predictive coding, a la Carl Friston, precision is actually kind of mathematically equated to attentional capture. However, in many experiments, we see almost the opposite, in the sense that predictable patterns are easier to ignore than random patterns. They do not attract attention. And we can see that behaviorally in experiments where we ask people to actively ignore regular or random patterns. So in this example, we have a participant listening in one ear to a difficult auditory detection task, which I will not describe. And in the other ear, they're listening to either of these random or regular tone patterns. They're entirely irrelevant. They are told to ignore them and focus on the ear that has the task that they have to perform. But their performance on this task depends systematically on whether the masker or the distractor was random or regular. And this is shown here. So the two conditions of CA and CD are different listening conditions. You can focus on one of them because the patterns are identical. In both cases, we see better performance and faster reaction times when the distractor is regular relative to when it's random. So suggesting that regular patterns are we discover them, we use the information about predictability to help us suppress them, but they don't attract attention. Another way in which we can sort of ask the same question is by looking at the eyes. And this is a recent study from Alice Milne that has used the pupil response as a measure of attentional capture. So we know that pupil responses can reflect attentional capture and attentional engagement. And so Alice, in her experiment, she recorded pupil responses by playing sound sequences to her participants. And these were either regularly structured or random sequences. And she used both these deterministic regular versus random patterns that I've shown you in the beginning, or saffron type sequences like the ones that Lucia was talking about, where people were pre-exposed to this artificial language and then were presented with sequences that are either consist of words in that language or not. And the prediction was that if regular patterns attract attention, then we should see enhanced sustained pupil responses to these regular versus random patterns. But if the presence of predictable structure facilitates processing, helps you conserve resources, and we should see a reduced sustained pupil responses to predictable relative to unpredictable sequences. And this is exactly what Alice sees. So what is shown in this picture is time. So it goes from 0 to 8 seconds. Each sequence was 8 seconds long. And what this is showing is sort of normalized pupil diameter. And so the red trace here are the random sequences, and the blue trace are these in fact, these are the saffron sequences. And you can see that the blue one, they show sort of a similar rise initially when the sequence starts, but then the blue one shows a much faster decline, which we interpret as the fact that discovering that this is regular helps you conserve resources. How much time do I have left? Actually, I lost track of how far behind we are.
We're not very much far behind. I think you can have another 5 minutes, Maria.
Okay, excellent. So this is the last part of my talk, and one that actually relates to what we discussed before about memory and predictability. And so the question that we were seeking to ask in this study is, I discussed before these stimuli that contain transitions from random to regular structure. And for you guys, for our subjects, for the brain to be able to discover the emergence of this regularity, it needs to have had some sort of buffer, some sort of memory buffer that maintains the information that it has encountered in the kind of preceding few moments, to compare the information that it's currently receiving and then decide whether the stimulus is repeating or not. And the question was, is this memory store that we're using when we're scanning sequences in this way, the memory store that helped us observe these changes to these EEG or MEG sustained responses, is this memory store encapsulated? So is it just used momentarily for whilst you're scanning and then deleted, or is it retained long-term? And to do that, Roberta Bianco in my lab has conducted a heroic series of experiments where what she had people do is she had them just detect the emergence of these regular patterns, so like the ones that I've played you before. So half the stimuli were random, and half of the stimuli had these transitions from random to regular structure. So they sound like this. So these are a bit harder than before because each pattern consists of 20 different tones. And so the duration of each pattern is 1 second. And they also heard these sort of step stimuli as a way of controlling for their reaction time. I think there might be another sound. Yeah. Sorry, no more. So participants were doing that. They were just detecting these transitions, but unbeknownst to them, three different regular patterns reoccurred throughout the presentation, so every about 3 minutes. So the random patterns were always new, the kind of leading random patterns were always new, but just occasionally these regular structures reoccurred. And kind of to demonstrate this more clearly, so we had these blocks. Each block was like 8 minutes long, and there were a bunch of random trials. And there were a bunch of trials where people had to, detect this regular structure. And unbeknownst to participants, a few of these reoccurred every 3 minutes. And there were kind of 3 different ones that reoccurred. And the idea was that if people are remembering this information that they've encountered in some sort of memory store, then when they re-encounter a pattern that they've heard already, their reaction time should be faster. And by tracking this reaction time and looking at how different it is between these reoccurring and novel sequences, we can determine sort of these latent parameters that are governing what and how we remember. And so these are the data. This is just D prime performance to show that people were really good at this task. So D primes are always above 3, people can do it. Our measure is reaction time. And this is shown in this pattern. So the blue traces are reaction times to these novel patterns. And as you can see, there's a bit of a learning effect, and then it stabilizes. For recurring patterns, which is this yellow trace, we see a really rapid emergence of a memory effect. So this reaction time advantage. So it starts out of, so each block is 3 presentations, 3 minutes apart. So after, already after 3 presentations, you have these memory effects that then stabilize over time. And remarkably, this memory effect lasts after 24 hours and after 7 weeks. So people are, despite these stimuli being completely meaningless and not relevant to our life, they seem to somehow stick in the brains of our listeners for, for all intents and purposes, forever. This is just showing you the distribution of participants in block 1, block 5, showing that, all of them are now below. So being below the diagonal suggests that they are exhibiting this memory effect, and it persists. So we have an attrition, so fewer participants after 7 weeks, but it's there all the time. And with I don't have time to go into this, but we have a statistical learning model that we've been employing to understand performance in these stimuli kind of more generally. And we can adapt it to try to look at what memory constraints might reflect or support this performance. And it suggests that, the key model parameters that explain human performance well are a short-term memory half-life of around 3 seconds and a long-term memory half-life of 500 seconds. So suggesting that people are able to maintain, irrelevant and sort of random auditory information in memory for quite a long time. So yeah, this is kind of the conclusion of this, that humans have a remarkable capacity to gather and retain information that they have rarely encountered in complex scenes. And this happens even when sound sequencers are featureless and irrelevant. And we think that the processes that support this are probably the same ones that play a role in helping us extract information from language and so on. And so my broader summary for all of this is that the auditory brain is awesome. It tracks and remembers the statistics of unfolding sequences, even when they're not behaviorally relevant. And the network that supports this encompasses the standard MMN network, plus the hippocampus and frontal areas for model violations. And more generally, my spiel is to try to sort of convince you that these types of rapid structured sound sequences are a very useful tool with which to study the brain as a statistical learning machine, because it's a flexible system that allows us to impose all kinds of regularities. And yeah, thank you very much. And thank you for the people who did all the work.
Thanks a lot, Maria. Thank you. Questions? I see that there's one raised hand.
Maybe I I should unshare so that I can.
Francois, there we go. You're free to talk now.
Yes, thank you very much for this nice talk. Yes, my question is related to your experiment where you compare a first sequence of random sounds with 20 different sounds. And then the sequence moves to more regular random sequence, which has 10 sounds.
Yeah.
So we move from 20 random to 10 random. And if I understood well, the idea of your Bayesian model, then there is I'm getting lost here. Yes, so in the Bayesian model, the model is tracking the precision of this pattern, and so my question is, if there are 10 random sounds, so I guess the precision is the variation, the variance across at these 10 levels, at these 10 levels of sound, there is a variance, and what you say is that this the brain is following the precision at each of these 10 layers, or these 10 levels of sounds, and how is it possible? Because I don't know, working memory is limited, and 10 levels is quite a lot for the brains. Do you really think that the brain is able to kind of do the average of this variance at each of these 10 levels, because it needs to remember the average value of these 10 levels? Sorry, so, I don't know if it's clear enough, but
so I think it's partly so firstly, the auditory system itself is already channeling. So one of the first stages of hearing involves filtering the sound into, different frequency channels. And so in fact, the finding here that the default, the model where the default listening state is channeled is in that sense not surprising, because we know that happens anyway. However, I mean, I didn't show, when I showed the model schematic, I showed kind of an abstract model output. But for these type of sequences where you go from a distribution that covers 20 tones to a distribution that's centered on the middle frequency range, we expect it to, the predictive distribution to shift to a distribution that is more centered on the center frequency. So we expect the sort of the initial, let me see if I have, if it's drawn sufficiently clearly in the representation of the model. Sorry, I should just go here. It will, so the predictive distribution over learning will reshape itself to be more similar to the distribution of the signal that you're encountering. Unfortunately, the example here is a little bit different. So this is for a stimulus that ends up having two concurrent channels like this, and where you can see that the prior sort of begins with the sort of even weights across all channels and then reshapes itself to match the distribution of the signal. Similarly, I mean, we expect for the stimulus that I've shown, the distribution to reshape itself to be more weighted towards center channels. I don't know if that kind of answers your question.
Yes, thank you.
Thank you, Maria. There's Floris with a question now.
Hi, Maria. Great talk. Really enjoyed this. So I was curious about the attentional capture that you were discussing. So I thought it was interesting that indeed you used the data by Milne, I think it was, showing that for regular sequences, you basically need less resources, right? As it basically you need less attention for that.
Yeah.
But then at the same time, when the beginning of your talk, when you are comparing these kind of regular and random sequences as you were just hearing them for the first time, there it looks like there's really extra processing dedicated to the regular sequences, right? Where you get your auditory cortices, but also some other areas that maybe some people would even associate with some form of attention that become more active for regular than for random. So I was wondering if there's maybe, rather than regularities being more easily ignored, that there's a bit of a push and pull, that in the beginning, when you encounter regularities, you actually attend to them more to learn them. But especially if they're simple, once you have learned them, then you can disengage and your attentional system becomes less engaged. Is that, you think, what you're seeing, or is there something else?
I don't think that that's what we're seeing. I was kind of, as you were talking, I was thinking whether I have the right slide for that. So here in this data that I have shown, these are saffron sequences. The people were pre-exposed to them before we measured what is shown here. But in the same experiment, Alice also used novel, regular versus random patterns, run-of-the-mill deterministic type sequences like the ones that we've been using in the past. And the pattern of pupil responses is identical. So nowhere do we see more responses to these regular ones relative to random ones. My hypothesis is that random ones always require more attention because you're constantly, you're processing them more, partly because you are primed to try to make sense of them. So the energy that you devote to trying to find regularity is actually expressed when you're processing random sequences.
Right, but how do you match that then with the beginning of your talk, where you show like extra resources for the regular compared to the random? So
this is a big mystery for us. So firstly, I think that potentially this large these are huge changes in brain activity. They're not excitatory. I think that they either reflect increase in inhibition, which would be then consistent with the behavioral and the pupil responses. It would still mean that you're devoting more energy, but you're devoting more energy at a low sensory level that helps you then free up the energy that's associated with the bottom-up driven attentional capture. So that's kind of one working hypothesis. The problem is it's hard with human brain imaging, so EEG, MEG, or fMRI, to investigate, because bold responses and EEG responses, you can't distinguish inhibitory from excitatory activity. So our friends doing animal work might we might need their help to help us figure this out. Another approach is actually related to Vadim Nikulin's recent hypotheses about non-zero centered oscillatory activity, which maybe is not the time to talk about now, but I'm happy to talk about later. Yeah. But I think, it's basically not excitatory.
Okay, well, interesting. Thanks.
There is another question from the attendees. So Lucia, if you forgive me, I think I would give priority to that, because we also have the round table next, so we surely have time again to debate. And by the way, I don't think there's a real boundary between the two things. So we can sort of gently move on to that phase where we try to get more general and sort of put what we've heard today sort of together. So Yudajit, you can talk now.
Thank you very much. I really like to talk. I would like to ask, just to clear up what I was, what was being just discussed in the previous question. If I understand correctly, the way Carl Friston's the predictive coding model works, like Bayesian prediction, is the hypothesis that the speaker presents is that it's like the predictive top-down signal which is inhibitory, which is leading to less attention devoted to trying to figure out the regularities, because now it's predictable.
So actually, I think that, by the way, I should apologize. There's a helicopter that has positioned itself above our house for the last, 15 minutes. I don't think that our findings are consistent with the strict Friston predictions, at least as they've been laid out in some of the kind of publications about predictive coding and attention. So it's difficult for me to, because Friston, they have, for example, kind of equated precision with attentional capture, almost mathematically. So I have a difficulty resolving although I think when I talk to Carl, he has a very clear explanation of our findings within his theory, but I just don't understand them. Sorry, I can't be more helpful.
Like I was wondering if like precision was the point of like the attentional capture, rather like the yeah, like whether we were being able to predict it properly, like whether there was precision enough to like to have a statistic, like whether there was a statistical regularity present to like give enough weight to the precision. Like, so yeah, I am also not very clear, like this question. Yeah.
So I think precision is used in slightly different ways. I mean, you can talk about precision of stimulus, precision. I mean, so I talk about this in the context of the precision of your internal model. So how in the context of the model that I would have shown, it would be how narrow the predictive distribution is. So how precisely you can predict an upcoming sound. So the idea would be when the sequence is unpredictable, the distribution is going to be broad and the precision is going to be low.
Right, the model is not fixed, kind of like.
Wait, it can fit well. I mean, but the world can be unpredictable too. So in the random, the fully random case, your precision is low because your model could be perfect, but yeah.
Okay. Thank you. Yeah.
Thank you, Rajit.